I play the bass. But it is loud, hard, rock and party music.
What is it?
Okay, starting on page 13. So get your lecture packets out to page 15.
Okay, that's DNA replication?
All right, last time we finished talking about how cells use the three different links for the three nutrients to make ATP. Right? I built it on monosaccharides first, and then I plugged in these other two, correct? Everybody on that page?
Yes.
We only have, after today, one more lecture left on lecture exam 3, and then 2 weeks from today is our first lecture exam. Okay? So it's coming up. That was one big topic for lecture exam 1 as far as points goes. We covered it Monday. I'm starting now on the second big topic for lecture exam 1, and that's DNA, which is one of the two nucleic acids in our cells. Okay? Are we ready to do this? There are two subtopics under DNA. One is protein synthesis, I'm covering today. Next is DNA replication and cell division, I'm going to cover on Monday. And then we're done. Any questions? Any questions? Stay up with me now. First of all, you need to know what a nucleotide is. A nucleotide is a link to the chains we call nucleic acids. So it says up there. A nucleotide has three things. A nitrogenous base, I did it in reverse order. A sugar, and at least one phosphate group. That's why ATP qualifies, even though it has three, because it has at least one. Now the sugar names nucleic acid. For DNA, the sugar is deoxyribose. And for RNA, this right here, this H is an OH, has one more oxygen. Okay? And that is why ATP is an RNA nucleotide, because its sugar is ribose. But today we're going to talk about DNA, and the sugar is deoxyribose. Okay? Any questions? Here are the 4 DNA nitrogenous bases, and they are different than in RNA. Well, one is different. 3 are the same. Now these nitrogenous bases could either be large, 2 rings, or small, single rings. The large ones are adenine and guanine. Adenine is the base for ATP. That's where the A comes from. These two are the same in both DNA and RNA. For the single ring pyrimidines, you have cytosine, and that's also the same in RNA. But thymine is only in DNA. In fact, you might want to write that next to thymine. Only in DNA. It's not found in RNA. Easiest way to remember this, because you do also need to be able to visually recognize the difference between purines and pyrimidines. The shorter word is the bigger structure. The shorter word purine is double ringed. Pyrimidine is a bigger word. It's single. You must be able to visually recognize purine and pyrimidines. You don't have to recognize the 4 individual bases. Just the difference between purine and pyrimidine. Okay? This from your textbook shows both DNA and RNA nucleotides. And here's the difference. In DNA you have thymine, in RNA you have uracil. So next to this figure, this is DNA only, and next to uracil, this is RNA only. but the rest of them are the same. And you can see here ribose, the difference. You just change this H to an OH, and now you have ribose. Everybody see that? Very similar sugars. All right, in our cells, DNA always exists as a double strand. They're held together by weak hydrogen bonds. Go ahead and fill in the blanks. And you can see some of the chemical structures of these on page 14 in your lecture packet. You can see on the top of page 14 the nucleotide with the three things. And then the five-sided ring sugars, deoxyribose and ribose, and then all the bases. Again, you don't need to memorize all the 5 bases, just be able to recognize the difference between a purine and a pyrimidine. Okay, all right, everybody got these blanks? Look at this picture with a critical eye. Are these strands mirror images of each other? Is there a left strand mirrored by a right strand? Is there a left strand mirrored by a right strand? No, they're all left-handed. This strand is going like this. This strand is going like this. Can you see that? They're all left-handed. They are oftentimes numbered by the carbon number in the deoxyribose, by the 3 and the 5, don't worry about that. Also notice the hydrogen bonds is holding the double strand together. Now, in a double helix, it looks like a spiral staircase. In this figure, they went ahead and straightened it out, it looks like a ladder. You see that? What are the vertical runners made up of? Spit it out so we can get through this. If I don't have time, I'm not gonna be able to go over the green sheet. Vertical runners of this ladder are made up of what? Sugar, phosphate, alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's the vertical runners. Sugar, phosphate, sugar, phosphate, sugar, phosphate. Everybody see that? Notice that the pentose, the 5-sided point here, is pointed up on the strand, pointed down on the strand. That's how you can tell they're not mirror images. They're all left-handed. What are the steps of the ladder made up of? And this is the topic of the second bullet. The nitrogenous bases. 20 angstroms is enough for 3 rings. Notice each step is 3 rings. A purine matched to a pyrimidine. 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 3 rings. Then also notice, well, if you're building this ladder, this is a big board, this is a small board. These boards only have 2 bolts between them. Big board, small board, these have 3 bolts. So it's not only you gotta be the right size, you have to have the right number of hydrogen bonds, which makes this base pairing highly specific. A goes to T with 2 bonds, C goes to G with 3 bonds. That might be a test question. Here it is right here. Here you can really see how these are all left-handed. This one's right side up, this one's upside down. And here's those numbers for the number of the carbons in deoxyribose. It's either heads or tails to head. Everybody see that? But in real life, it's not a ladder, it's a spiral staircase, what we call a double helix. Now, each intact double helix is not by itself. It's also mixed with proteins. These proteins are called histones. But an intact double helix with its proteins, that is a chromosome. And the reason why this is not considered a nutrient is because in every cell, at the most, you only have 46 of them. You have trillions of 1, 2, and 3. You got 46 in number 4. Do you see why it's not a nutrient?
Mm-hmm.
When you take a picture of the chromosomes at metaphase, that's what's called the karyotype, we can actually see the 46, and we see that it comes in 23 pairs. So there are 23 types of chromosomes in our species, but you have 2 of each, one from mommy, one from daddy. But what is on each of these chromosomes are genes. Okay? These genes either code for proteins or they regulate those that code for proteins. On this bottom bullet, write a little note off to the side for this bottom bullet. Genes either code for proteins or they regulate those that code for proteins. So this is going to tie this, where the genes are on, codes for proteins. And that's on the genes. For example, the D antigen is a protein that is on our red blood cells. On chromosome number 1, some people have this. And if they do, they make the D antigen, and they are Rh positive. If you don't have this gene, well, not only that, both chromosomes number 1, neither one can have the gene. And if neither one has the gene, you are Rh negative. Everybody got that? If you're missing it, you don't make the D antigen, you are Rh negative. We're going to cover that when we talk about blood later. But it's coded right there on chromosome number 1. This is a picture showing you how it's not just a double helix, but also the proteins. The histones act, if the DNA double helix is thread, the histone proteins act like a spool. And the thread just wraps around the spool. Can you see that here? Can you see that there?
Yes.
All right. Back when I went to school, they used to think there was over 100,000 genes in our 46 chromosomes. And then eventually that number went down to about 50,000. And then eventually they did the Human Genome Project. Do you guys remember that from, let's see, that's almost 20 years ago now, maybe even more. Dang, time is flying. When they did the Human Genome Project, they said, oh my gosh, we're way off. There's maybe 20,000. That's what it says there, or 21,000. That might still be an overestimate. So out of our 46 chromosomes, there's only 46 strands, but some of them are really long. And out of the 46 chromosomes, we actually have 3 billion base pairs. Of those 3 billion base pairs, less than 2% does anything. Over 98% of your DNA is space filler. It's serving only one function and making you different from chimpanzees. It's just dummy space filler. Okay? But when we talk about protein synthesis, the gene that codes for proteins has to separate. Hydrogen bonds have to break. So you have two different strands once the double helix is separated, and only one half is the gene. The other half is not even the gene. But it is crucial, because next week on Monday, when I talk about DNA replication, say like if this is a double helix and it separates, and this is the gene, you need this strand to make the gene when the cell gets ready to divide. You following me? So it's not like it's not used at all. It is used, but the actual gene is only one half of the double helix. Are we clear? Are the words making sense?
Yes.
Make the words make sense. Everybody look at me. Make the words make sense in your brain. If you cannot, you must ask. Or come talk to me. But do not just let it sit there not making sense. You'll never be able to do pharmacology if you just let things not make sense. Okay, that's what this bullet is saying. Only one side of the double helix is the actual gene. But you do need the other side to reproduce the gene when you do DNA replication. So we're gonna go through this sequence. Again, everything is a sequence here, just like with metabolism. Do not be intimidated. Take a step back, see what this whole thing is used for, the context. Break it up into sequential little steps. What we are talking about with protein synthesis is how do we get a DNA nucleotide sequence to an amino acid sequence to make a protein. Write that down. Dude, this is how you're gonna understand, this is how this makes sense. Big picture. Write it down. Otherwise it's just content and details. The whole big picture of this topic on protein synthesis is how our cells go from a DNA nucleotide sequence to an amino acid sequence in a protein. The topic is protein synthesis. How do we make proteins? You gotta go from a DNA nucleotide sequence to an amino acid sequence in a new protein. And we're gonna go through all this thing that's in this picture plus some. Before we can do that, we have to talk about the 3 different subtypes of RNA. And that's the little letter in front of it. There are 3 subtypes of RNA. And this is a big picture of what I just described. This slide here is telling you the big picture. We're going to go from a nucleotide sequence to a specific amino acid sequence in a new protein. That's the big picture. The nucleotide sequence on your genes is called the code. We have to go from that to an RNA sequence, what's called the codon. That is going to be on messenger RNA, that's what the M stands for. Don't worry, I'm going to go in there in a little bit more detail. Then that's got to go to a transfer RNA, that's a little T, and that sequence is called an anticodon. But on the other side of the transfer RNA, so one side has the anticodon, the other side has the amino acid. And that's what's going to give you your amino acid sequence. Everybody got that? So we got to go from the DNA code to a messenger RNA codon to a transfer RNA anticodon to an amino acid sequence. Look at your essays. Look at your essays right now. I know we've covered all the essays, but on the very first essay,
Protein structures.
everybody got the essays out? What is primary protein structure?
Amino acid sequence.
Amino acid sequence. That is what we're actually talking about here. When we're talking about protein synthesis. We're not even going to talk about how we get secondary, tertiary, and quaternary. We're only talking about primary protein structure, amino acid sequence. Okay? That's what's on the screen. This is the big picture. We are going to go from a DNA nucleotide sequence in the code eventually to an amino acid sequence, which is primary protein structure. Everybody got the big picture?
Mm-hmm.
Okay. So let's go over the differences between DNA and RNA. The obvious one is that it's got a different sugar, and you can tell because it's got a different name. The sugar has one more oxygen in it, so it's ribose. Also, while DNA is always double-stranded, in your cells, RNA is always single-stranded. The other one has to do with that one nucleotide, that one nitrogenous base that's different. Well, the two purines, adenine and guanine, A and G, are the same, and the one pyrimidine cytosine is the same. The bottom diamond here is showing you that in RNA, there's no thymine. Instead, it has a very similar pyrimidine called uracil. This is one example of specificity. They're similar, they're not the same. Okay? All you gotta do is just change the T's to U's, and you're an RNA. So here it is, so it's ribose, and we're gonna cross off thymine and replace with uracil, and it's always single-stranded. Okay? Are we good with the differences?
Yes.
Now let's go over the three different types of RNA. This is the same figure that was one on the previous slide, same figure. Messenger RNA is what is copied off the gene. That's what has the codons. It is the product of transcription. It's going to be important to remember C goes to G with 3 hydrogen bonds, because that's the same in DNA. But since there are no thymine, no T's in RNA, A goes to U with 2 hydrogen bonds. Very similar to thymine, it's just not thymine. Transfer RNA, again, has the anticodon on one side and the amino acid on the other side, like that. Anticodon on one side, amino acid, in this case aspartate, on the other side. But protein synthesis is done by ribosomes. Ribosomes, remember from anatomy, is the organelle of protein synthesis. It is actually the perfect organelle if you're doing the biological hierarchy of organization, to go from atoms, molecules, macromolecules to organelles, because all a ribosome is 2 boulders. A large boulder of rRNA and protein and a small boulder, and they clamp together. Ribosomes are made up of rRNA and protein. they are the smallest organelles, because all they are two folders of macromolecules. Okay? So here's the three types of RNA, and you can see it in the first small case letter. Any questions? Again, the product of transcription is messenger RNA. Here's a transfer RNA that has the anticodon on one side and the amino acid attaches on the tail end of this end of the chain. It is still single-stranded, but there are sections where it will double strand on itself, but it's still only one strand. Everybody see that?
Mm-hmm.
And here's the two folders that make up a ribosome. Large and small. And they clamp together on the messenger RNA. So here's the sequence. Let's see. So say we start out with that gene. We're gonna do transcription first that happens in the nucleus. That's gonna produce a messenger RNA that goes out those large nuclear pores. This is the reason why the nuclear envelope has these large holes, because messenger RNA is big. And you gotta have these huge holes so that messenger RNA can go out to the cytoplasm, where it can clamp onto ribosomes. Or actually the ribosomes clamp onto the messenger RNA, and that's the rRNA, and that has sites to put in two transfer RNAs. It has two different sites. Now look at a transfer RNA that has nucleotides and one amino acid. What is the ratio of nucleotides to amino acids? 3:1. So hemoglobin is a quaternary protein. One of the globs in hemoglobin has 282 amino acids. Let's round it off to 300, just for illustration's sake. That one glob out of 4 in hemoglobin has 300 amino acids. How many nucleotides is in the gene for that hemoglobin?
100?
Yeah.
If there's 300 amino acids.
100, right? 3:1. No. Is there
3 to 1 in the opposite
300?
direction.
Oh.
It's 3 nucleotides. So if there are
So
300
900?
of these, there's 900 of these.
Okay.
Does everybody see that? It's a 3-to-1 ratio. Everybody following me?
Yeah.
All right, go ahead and fill in the blanks.
I think I'm following. Is that what these numbers are? 3:1. Where are you seeing the 3:1? Where does it say 3:1? Or or am I supposed to just know that?
Just
Anyone
look at
else?
it.
Where? Okay,
3 bases, 1 amino acids.
okay, thank you. Okay. Sorry.
What you need to know is transcription happens in the nucleus, it's underlined. The enzyme that's driving it, there's actually more enzymes than this, but the one you need to know for lecture exam 1 is RNA polymerase. Remember that the DNA has to unravel at that point, and it's only making a complementary strand on the gene. The other strand is just sitting off to the side. Once it hits what's called, you'll see here in a little bit, the terminator codon, it'll dislodge, float out the nuclear pores to go out in the cytoplasm. How do I know this is the sequence? Because A goes to T, U, remember there's no T's in RNA, only U's. U's goes to A's, G goes to C, C goes to G, A goes to T. Do you see how I can do that? Guess what? You're gonna have to do that on lecture exam 1 too. If I give you the gene, the code, you gotta give me the codons. Okay? Everybody got this? Look at this. What is this first one right here that's forming in?
ATP.
That's ATP. RNA nucleotide with 3 phosphate groups. This is ATP. Look at this animation with a critical eye. Every base pair that's coming in is a triphosphate. And they got the high energy phosphate groups with 7 kilocalories per mole from ATP. To make one of the boulders in hemoglobin will take 900 ATP. They're just going to transfer those high energy phosphate groups to these bases. Everybody got how this works? Here's the DNA gene. The other strand is sitting off to the side doing nothing. And just like with DNA replication, we'll see later, hydrogen bonds form first, then this covalent bond between nucleotide bases we call a phosphodiester bond, which you must also visually recognize for lecture exam 1. So this has,
Is that just the bonds in between the
Uh-huh.
ATP on it?
So if this here in proteins between amino acids are peptide bonds that you must visually recognize. Between this is phosphodiester bonds. They're the equivalent. But hydrogen bonds form first, so they make sure that the right base is on there. It's got to be the right size with the right number of hydrogen bonds. Once that's assured that it's the right base with the right number of hydrogen bonds and the right size, then the phosphodiester bond is formed. Hydrogen bonds first, phosphodiester bonds second. Hydrogen bonds first, phosphodiester bonds second. Phosphodiester bonds are strong because they're covalent bonds. Hydrogen bonds are weak.
Does this happen with DNA too, or just the RNA?
In fact, if you want to read ahead, don't.
I'm good.
It's on page 16. It looks
Okay.
very similar. Okay? And here's the enzyme RNA polymerase. RNA polymerase we'll look at here in a little bit at the end. It makes the DNA acceptable to be added onto with RNA. Okay. Now you might have thought this was random, but it wasn't. The reason why I started with TAC is I went backwards with complementary base pairing with this, which is the initiator codon. The initiator codon codes for an amino acid we call methionine. Go ahead and fill in the blanks there.
So is AUG always the start?
Uh-huh.
Okay.
And you know what that means? That means methionine is always the first amino acid. Well, it starts out that way anyway, but that's not the final protein. Very few final proteins actually have methionine as the first one. It just starts out that way. Okay, you know how I keep this thing straight between transcription and translation?
It's backwards, right?
You want to know? Maybe this would be helpful for you. Maybe not, I don't know. Transcription, probably the only transcription that I am usually aware of is what happens in court, where spoken English is transcribed into written English. It's still English. Okay? When you look at transcription here, it's going from a DNA nucleotide sequence to an RNA nucleotide sequence. It's still nucleotides. Do you see that parallel? But when we talk about translation, we're going to go from a nucleotide sequence to an amino acid sequence at a 3-to-1 ratio. It's a different language, like translating English into Japanese. That's translation. If it's still nucleotides, it's transcription. If you change from nucleotides to amino acids, that's translation. Everybody got it? All right, translation has 3 sequential steps, and they're underlined. You start with initiation. You have to start with this code, because that's complementary to AUG, which codes for methionine. Then, and watch this in animation, you'll see that when the ribosome clamps on it, it's 2 subunits, it has 2 sites. I think they're called A and P sites. And it just slides down the messenger RNA. The red lines here are the peptide bonds. This adding amino acids is elongation, the second bullet or diamond. And again, it's at the 3-to-1 ratio. There's 3 nucleotides for every amino acid. So what happens in elongation, it is going to break this covalent bond. When it forms this covalent bond is a peptide bond between NCCs. Then the ribosome is going to shift 3 nucleotides down. That's going to break this bond, and it's going to form this bond. then the ribosome is gonna shift 3 nucleotides. Then it's gonna break this bond, and it's gonna form this bond. And it's gonna keep on going until it hits one of 3 terminator codons. You don't need to know all the codons, but you do need to know those 4. The 1 initiator codon and the 3 terminator codons. And at the terminator codon, the 2 subunits of the ribosome fall off. The messenger RNA is free floating, it'll be degraded by enzymes. And you got your new protein. But you went from a nucleotide sequence to an amino acid sequence. That's translation that happens in the cytoplasm.
When you say any of those 3 codons will stop the process, any of
those 3.
anything ending in UAA or UAG or UGA?
Uh-huh.
Okay.
And all you have to do, now remember, codons, everybody look at me, codons are on the messenger RNA. Everybody got it? So which means you could take any of those 3 terminator codons and backtrack it to whether it will be in DNA, and that's when the whole thing stops with the messenger RNA.
Okay. So not in translation, it's in.
But yes, when you're talking codons, it's on the messenger RNA.
Okay.
Everybody got it? Okay, another thing to think about here, that hopefully it'll be easier because at least one Scantron question is going to be, can you do complementary base pairing between DNA and RNA? Is that if you think about complementary base pairing, it's like taking a negative in nucleotides. Correct? Correct?
What do you mean?
A negative. It's the opposite,
Oh. Oh, yeah. Okay.
right?
Uh-huh.
Well, if you look at the anticodon, if it's going to do complementary base pairing, it's a negative of a negative. And what is a negative of a negative?
Positive.
Positive. So even though there's A's, let's go to one that doesn't have it, it's not going to have any U's in it. Look at this third one. ACG, a negative of a negative, is ACG.
Wow.
Is that creepy guy out there?
Creepy guy.
Yeah, there's a creepy guy that comes out here. I can still close the door if you want. Oh, yeah, if you see. Yeah, I swear, this is God. There's sometimes I think he's following me. It's just really creepy.
You have a fanboy?
We'll give him a finger.
I got other more important shit to think about. All right? Any questions on this? Everybody look at me. This is way more complicated than what I'm giving you. If you think this is complicated, you really gotta readjust your view of the world. This is nothing. I'm giving you just the bare minimum. If I don't give you at least this, you're not gonna even be prepared for the TEAS test. You gotta at least have this. Any questions? So this is from your textbook showing the same thing. Initiator codon codes for methionine. The anticodon is UAC. Here's the P and the A site. First one is the P site. Then when the second amino acid comes on, the A site, it's going to break these covalent bonds here and form a new covalent bond we call a peptide bond. Once the peptide bond is formed, the ribosome shifts 3 nucleotides down the messenger RNA. A new one, the third one, will come in and occupy the A site. This covalent bond will break, and the next one will form. Then it shifts 3 more, that's elongation. Okay, so this is initiation. Maybe on this slide write elongation. and eventually it'll terminate. And now what I wrote here on purple right here is the ribosomes can be free floating in the cytoplasm. And if it's a long messenger RNA, several ribosomes can clamp onto the same messenger RNA. They call that a polyribosome. This is about as magnified as you're gonna get in an electron micrograph. You're actually seeing macromolecules. The horizontal line is messenger RNA. Each one of these boulders is a ribosome, our RNA and protein. And what you're seeing coming off of them are new polypeptides, proteins being built. You can actually see that in this picture. Okay. Or it could be in rough endoplasmic reticulum. Usually cells that have to make a ton of protein will have a lot of rough endoplasmic reticulum because it's covered with ribosomes. It forms it forms assembly lines for protein synthesis. So say the beta cells in your pancreatic islets that have to make insulin, another quaternary protein, those cells of beta cells are loaded with rough endoplasmic reticulum. Any questions?
So the protein structure, sorry, this isn't about that. The MET and the AA2 and the AA3, are those just the names for each?
Yeah, the only
protein
one that I
structure?
know for sure is methionine because I don't have the table of codons memorized.
So we don't need to know those?
No,
Okay.
except for the fact
The-
that the initiator codon is AUG and it codes for methionine.
Okay.
Okay? Okay, any questions? Any other questions? This is, again, as simplified as I could possibly make it. Now, there is some other little additions that I will add on to. And one of the things is like the Golgi apparatus or Golgi complex. It does finishing touches on proteins. So when we talk about this, we're talking about the initial proteins. And all the initial proteins all have methionine as the first amino acid, but after it goes through the Golgi complex, usually the first section of the protein is taken off, and then methionine is gone. So in all your regular proteins, methionine is rarely the first amino acid. Okay? Any questions? All right, so like, for example, in insulin, yes, proinsulin that's bubbled off the rough endoplasmic reticulum will have methionine as its first amino acid. By the time it goes through the Golgi complex, actual functional insulin no longer has methionine as the first one. Does that make sense?
Mm-hmm.
Everybody got that?
Yes.
Okay. All right, then let's get to the topic of gene regulation. Oh, I was supposed to remind you too, my son wanted me to remind you that tomorrow I'm in the STEM Center at 11 to noon. God, I better write that down or he's gonna give me shit. On my office door, I have a sign that talks about my Thursday office hour, but when I have anatomy lab exams I have to set up, I'm gonna be in the anatomy lab setting up the lab exams, so I won't be in either my office or the STEM Center. But I'll put a sign on my office door that says when I'm not gonna be in either. What is that, W22?
Mm-hmm.
Oh, my band is even above the STEM All right, gene regulation. So I said initially that the genes on all your chromosomes either code for proteins, and this would be one of them, or they regulate those that code for proteins. You guys remember that? Now we're going to talk about those that regulate those that code for proteins. And two different forms that you do have to know is positive control and negative control. One example of positive control is induction. Go ahead and fill in the blanks there.
Turned on.
Positive means it's turning on. So in this case, you got to start with a gene that's off, and then an activator is introduced that turns it on. Okay? In its simplest form, the activator bends the DNA into a shape that is favorable for RNA polymerase. Okay, just fits better. Any questions? Key here for this slide that I would write next to it is you start off and an activator turns it on. It's only going into the positive direction. Positive control, the gene is induced, so that's why it's induction. Okay, everybody got this? You do have to know this for lecture exam 1. You can see that it's much more complicated. Here showing one activator with RNA polymerase. There's actually several that have several regulator genes with several activators. It can be much more complicated. Then you also have to know this. This is a form of negative control called repression. To have repression, you have to have the gene starting off on. It is normally just going on. Then you're going to introduce a repressor molecule that turns it off. And in its most basic level, the repressor blocks RNA polymerase. So if you look at these figures on both slides, this one here, this basic figure, the gene that is being transcribed is in green. The yellow and the purple ones are regulator genes. You might want to write that down somewhere. The actual transcribed gene that codes for proteins, so there's two types of genes. Those that code for proteins, that's the green one. And those that regulate those that code for proteins, that's the yellow and the purple one. Everybody got it? Any questions?
Yellow and purple regulate and green code.
Is actually coding for proteins. It is being transcribed. The green is the one that's being transcribed. It's actually coding for proteins. That's the green one. So you see the same thing here on the repression slide. Same thing. So here the repressor binds to the operated gene and it blocks RNA polymerase. There's no transcription. Turns it off. The key here about repression is that it is more flexible. Because all you have to do is remove the repressor and it turns back on. This is a good on and off switch. This is the basis for the Lac-Operon theory of gene regulation, which is on page 17 of your lecture file. Lac-Operon theory, what is that? That's not even human. This is what has been discovered in bacteria, because bacterial protein synthesis and gene regulation is way simpler than ours. So it's not even this isn't even ours, but we're gonna use it so you can kind of get an idea why it is this stuff happens. Now everybody look at me. For lecture exam 1, the only thing I'm gonna ask you about this is that you have to know that the Lac-Operon theory of gene regulation is a form of repression, negative control. I'm gonna go over several slides that describe this in sequential summarized steps. That's still way more complicated than this. I am going to bring up one other thing at the end on cyclic AMP, but the rest of the next few slides, where I'm going over the Lac-Operon, I'm not gonna ask any questions. So, you can either take the prevailing mentality in this valley and just veg for a few minutes, or you can remain engaged and use it as an example of how it's important when you see a big concept that you not be intimidated. Because that doesn't help anything. Nor does vegging. So if you veg, you're not even going to take this example. Take a big picture look at what this whole thing is for, then break it down into sequential steps that are doable. Are we ready? The bottom line for the Lac-Operon theory of gene regulation is that this bacteria, this E. coli, does not want to waste ATP making proteins it doesn't need. That doesn't even sound scientific. That doesn't even sound complicated, because it's not. Always take a step back and see the simple reality of why you're studying it. An E. coli does not want to waste ATP making a protein it doesn't need. That's it. Everybody got it?
Mm-hmm.
All right, let's go through it. Like mitochondria, and this is one of the reasons why biologists think mitochondria used to be bacteria, this E. coli chromosome is just a single chromosome, and it's in a big ring of DNA. So all these genes are connected. The protein that it wants to make is beta-galactosidase, which breaks down lactose. The key here, what you're going to see, is that the E. coli will not make beta-galactosidase unless lactose is present, because it doesn't want to waste energy making this digestive enzyme when it doesn't need it. Are we all on the same page on that? Okay. So, the operon is a series of genes, and there are 3 genes right in a row that are actually transcribed. This is a little bit different from other things, but it's going to have a single messenger RNA molecule that gets broken up into 3 different sections for 3 different proteins. In front of it, though, is the operator genes. And normally, there is a protein, a repressor protein, that binds to these promoter genes and the operator genes, which is in front of, and they call it the structural genes. Structural genes are what's actually being transcribed. Okay? Well, it's just sitting there blocking it. So this gene is turned off. So far, so good? Notice how there's no blanks, right? All right, so if lactose is introduced and goes into the E. coli, the lactose will actually bind to the repressor protein. It's a disaccharide, that's why it's showing you two colors, red and yellow. When it binds to these binding sites on the protein changes shape, and that shape now causes it to be dislodged from the promoter gene, an operator gene. Now that it's not blocking the genes, now RNA polymerase can just come on in and start transcribing. And remember, transcription, each one of these guys is a triphosphate. Each one of these guys took an ATP. So it's going to start burning up cell energy to make this messenger RNA. It's going to do it on all three of them. But the first protein is the beta-galactosidase. This beta-galactosidase is a digestive enzyme to digest lactose in the monosaccharides. And now this E. coli can make ATP from the monosaccharides, because it's got to get all the way to a single link to make ATP. You guys remember from Monday?
Mm-hmm.
Well, once all the lactose gets digested, the lactose that's on the repressor molecule falls off so it can be digestive, and now this repressor protein goes back to its original shape. When it goes back to the original shape, it's got great affinity for the promoter and operator genes. It binds to it, boom, turns this whole machine off. So let's go back and think about the big picture. An E. coli does not want to waste ATP, because each one of these is a triphosphate. Does not want to waste ATP making a protein it doesn't need. When lactose is there and it makes beta-galactosidase, it's going to make way more ATP than it took to make this protein, because it's going to digest the two monosaccharides and make a ton of ATP out of it. It's worth it to the E. coli to spend a little to get more. But as soon as the lactose is gone, boom, that cell will not waste any more ATP making a protein it doesn't need. Does the lac operon now make sense to you? What did I do? I took a step back, saw the big picture, what it's for, broke it down into steps. Which after, when you're done with me, is what you want to do. Okay? This is, of course, an oversimplification, like everything else I'm teaching you. And this is one thing you do have to know about it. Now go ahead and fill in the, are there any blanks on the script? Okay, the reason why I want you to know this is because of this molecule. And that's what I'll put over here. So if we have adenine, ribose, and 3 phosphate groups, someone tell me what this is.
ATP.
It's ATP. Now what is it?
ADP.
ADP. Now what is it? AMP. Adenosine monophosphate, 1. Everybody got it? But it only has one bond to the ribose. The phosphate group only has one bond to the ribose. Through this enzyme called cyclase. it'll form 2 bonds to the ribose. And when it forms 2 bonds to the ribose, it forms a circle of covalent bonds. Everybody see the blue circle?
Mhm.
This is cyclic AMP.
I've heard that before.
Now in your body, you have hormones that travel your whole body to signal other cells. And neurons will cross over with neurotransmitters in a small little gap called the synaptic cleft. Cyclic AMP is a signaling molecule for inside the cell. It never leaves the cell, but it signals the cell that something's up or something needs to be done. And we're going to see it again in the semester, so I want you to get it now. Signaling ligand, but it's inside the cell. And in the case of the E. coli, even though it's going to invest some ATP to make beta-galactosidase, it would actually prefer glucose over lactose. So, if glucose is present, the cell knows, you know what, I'm going to make the enzyme to digest glucose because I get more ATP from it. So the cyclic AMP levels stay low. But if there's no glucose and it's like, well, now I'm going to eat the lactose, but then the cyclic AMP levels rise and it knows it wants to make more beta-galactosidase. Does this make sense? I want you to know this because, well, you got to know for the TEAS test. You got to know what the cyclic AMP is. You got to know the enzyme, and you got to know what it's for. It's to signal the cell that to make the right decision. Okay? Any questions? See how this is ATP, and it is going to go ahead and get 7 kilocalories per mole and break this off. Then it's going to get 7 kilocalories per mole and break this off, and then it's got to invest a little bit of energy to put that second bond. And right here where I'm moving the arrow, that's the circle of bonds. Everybody see that?
Mhm.
And it's just gonna help this E. coli make the right decision. Again, you know, there's some good animations. I'm not gonna ask any questions on the actual steps of the lac operon. You just gotta know that it is a form of repression, negative control. And you do have to know about cyclic AMP. What it's for. Any questions? Do you guys want me to go over the green sheet? My anatomy students have already gone over it.